All right, here we go. Welcome back from spring break. Uh, we are working our way through all of units 1 through 6, I believe, um, of all the AP biology units. And in the last unit, unit 2, it was about cell membranes mostly, right? Because if you want to be alive, you need a body. You need to control what comes in and out of your body. You need to make sections and spaces for separate chemical reactions to happen away from each other. You need places to put enzymes and all these things that cell membranes do for cells, right? But the other thing that you need if you want to be alive is energy, right? And so energy is a big part of being alive. And as long as you have energy, you should stay alive. And when the energy runs out, then you won't stay alive anymore. And so we want to see how energy is used by living things and how do you capture it and how do you utilize it. Um, and so what that comes down to is chemical reactions, right? This is even going back to unit 1 now about all the different macromolecules and polymers and monomers and the way that we build molecules. And while there are a whole lot of different chemical reactions, they can really be boiled down to 2 different categories that are shown here. We have our hydrolysis reactions, where we're going to take things apart, and then on the right, we have our dehydration synthesis, where we're going to combine things together. We're going to add parts to the molecule. We're going to take parts away from the molecule, right? We're going to build and we're going to break things. And when you take all of this together, it is what you call metabolism, which is a key element of being alive. Um, and so this led us to having to do a little bit of talking about some math, although we don't have to really get into the math, right? But some of the big ideas are that chemical bonds store energy, right? You have to add energy when you want to make a bond, and if you break a bond, you'll get some energy out. There's a whole lot of math that can go into that that you do in chemistry class sometimes, and we did a little bit of that in class. I think, I don't know, it was so long ago now, it's hard to remember. But the point is that we don't have to do the math on this. What it comes down to is, do we want to add energy to make a reaction happen, or do we get energy out? So that's the graphs over here on the right-hand side. On the top, this is called an endergonic reaction, which for our purposes is pretty much the same as an endothermic reaction. We did a little bit of a thing where we learned that those aren't technically the same, but for our purposes now, they are the same, right? Something that is going to require the input of energy. It's going to absorb energy. Or a reaction that is going to release energy in an exergonic or exothermic reaction, right? Now, the big thing about this is that the energy is really in a, the reason why we have to add energy has to do with something else in this formula. So energy is this one called enthalpy. Again, you shouldn't have to ever say that word or read about that word, but energy is how much energy is involved in the reaction. Entropy is is chaos. It is disorder. And so the reason that you have to add energy in this first reaction is that if you think about the fact that we have 6 carbon dioxide molecules just floating around, they can float around and slam into each other and be as chaotic as they can possibly be. But if we want to put those 6 carbon dioxide molecules into one glucose molecule, we have to expend energy to do that. We have to lower the chaos. We have to build structures, and that requires energy. If we then eventually, oh, and the red on the orange isn't doing so great, but hopefully you can see my terrible glucose molecule. If we take that one glucose molecule and then break it into 6 CO2 molecules, that's going to raise the entropy, right? And when you make chaos, then you can get energy back, right? And so that's the basic idea, is that life uses energy to overcome entropy. And so these are the things that kind of govern the chemical reactions that happen in our bodies. And you don't have to go much farther than that. But there is an interesting thing about these molecules is that your biological molecules should be very stable, right? Like it would be kind of difficult to be alive if you were constantly worrying about your molecules falling apart. So macromolecules, lipids and carbs, and carbohydrates, and of course nucleic acids, these things are held together by very strong, stable covalent bonds. This means that no matter whether this is an exothermic or endothermic reaction, we need to add energy at the beginning, something called the activation energy, right? Again, this doesn't change the end energy. It doesn't determine whether it's being absorbed or released. You always have to add a little bit of energy at first to initially get everything happen, right? And so this is the part that makes life difficult when you think about it on a purely chemical standpoint, is that how do we keep getting over these barriers? And this is with something called an enzyme, right? And so in this graph, this line here, the pink line is showing the activation energy normally, but the blue line is the activation energy when you have an enzyme, which is a biological catalyst, and its job is to lower the activation energy. It doesn't change the final part. It doesn't change anything about the initial part. It only affects the activation energy. And that means that enzymes can make reactions happen that wouldn't normally happen, at least not on a timeline fast enough for them to be biologically useful, right? And so enzymes and energy are very important. They go kind of together, right? And so we then went and talked about enzymes. I'm going to start on picture 6 of this particular image, right? We have all kinds of other images you can find on your notes or in the old PowerPoints, but this one sums it up pretty well, that you have this enzyme, a protein that has a very particular shape. And the number one part of the shape that's important is the active site. It's the space where all the action is going to happen, the active site. The active site is where the molecules that go in are going to be found, called substrates. That's an important vocabulary word, right? And they're going to fit into the active site. Now, a big part of the AP curriculum is the idea that this is called the induced fit model, which means that the substrates don't just fit in. When they go in, the enzyme actually like changes to get more a closer fit, an induced fit. If you look in this image, you can see that the shape of the purple protein here is not the same as it is here, right? And so when the substrates fit in there, the enzyme clamps down, and then the action happens. You don't have to explain the action, right? But through various chemical means, there is going to be a lowering of activation energy, and out pop your products, right? One of the most important things about enzymes is that they go back to their original state. They are reusable up to a point. I'm sure they get worn out just from like physical wear and tear, but you can use them over and over and over and over again. They are not part of the reaction. They make the reactions happen. Now, that's a huge part of what could be on the test, right? Some more specific things that might show up here and there, again, it's hard to tell how detailed they're going to get, is that inside of the active site, there's usually these things called cofactors. And it's important about cofactors is that they are inorganic. These are metals. These are rocks, right? When you hear about vitamins and minerals, and I'll get to vitamins in a second, that's the minerals part, right? You need trace amounts of weird stuff like iron, right? Like it's kind of strange to think about, but without enough iron, then you can't put the iron into your hemoglobin. You need metal in your blood to hold the oxygen. Magnesium is, again, a rock, technically a metal. And it's in the chlorophyll, and it's going to do help us with the chemistry, right? Like one of the reasons why metals are not living is that they are more reactive in certain senses, right? And so we use them inside the active sites to get things done. A coenzyme is what we would call a vitamin, and it's important. The part about this is that they're organic, that you gotta get these from other living things, right? And usually that means it's going to be a plant. there are some vitamins that animals can make, but plants can make all of them. And so this is one of the reasons why you got to eat your vegetables, right? Coenzymes, vitamins, these are going to be molecules that usually donate a part of them, right? Like you eat vitamin A and your body's going to take the vitamin A apart and use part of it in one of the reactions, right? And the enzyme's going to take it off and add it to some other molecule. And so this is why vitamins are important. You have to reach a certain threshold, right? If you drop below a threshold, you don't have enough of these helper molecules to get anything done. And so this is why you got to have those important vitamins, right? So this is how the enzymes are going to work inside the active site. Now, the active site leads us to controlling the enzyme, right? Once we got past the basic structure, it's like, how do we make them work faster? Or not usually work faster. It's we're trying to make them work slower by adding a competitive inhibitor, something that is going to fit into the active site in the place of the substrate, but is not the actual substrate. Therefore, the enzyme can't do its job, and it just sits there all clogged up, and now you can't do any reactions. This is not the same thing as a non-competitive inhibitor, also known as an allosteric inhibitor, which is going to connect to a different part of the enzyme, which is going to make the whole thing change slightly and shift and make, in this case, you can see like the little horns have like moved inwards, and so now the substrate can't fit into it, right? If your active site is the wrong shape or blocked, then you aren't going to get any activity from your enzyme, right? And so this is the way that biological systems use to control their enzymes most often than not, something called a negative feedback loop. In this diagram, it illustrates something else that's going to come up in the next video, is that it's important to remember that usually enzymes come in like these long chains that have all related enzymes, and they all do one very specific small step. It might not seem like a lot of changing this molecule into this molecule. And maybe if you were a human building with Legos and you were able to take all the Legos apart and then reassemble them, then that would be easy. But you can't do that in your body. You can't literally take all the molecules all the way apart down to their atoms and then put them back where you want them. You have to just slowly change it, right? So think of this more like trying to change the parts of an airplane while you're flying. Like you can't just take the engine apart, but you could change this one little thing here. And then once you get that done, then you can change something else and you can slowly get it to happen, right? And so in this particular case, the final product that we're going to make is going to start to build up. And when you have enough of that product, the concentration will be great enough that it will increase the probability that it will come in contact with this enzyme, which is enzyme 1, right? Let's make sure that we realize that in this diagram, this thing right here is the same as that one. It's enzyme number 1. It's the beginning of the whole chain of enzymes, but now the allosteric inhibitor has made it change its shape, and now it can't do this part, which means this guy can't do its job and that can't do its job, and then you don't get any of this product. But once you don't have any product, your body starts to use it, and then this falls off and everything starts up again. And again, this is called a negative feedback loop, and this will come up again in some of the other videos if you remember. Biology uses this frequently, right? But this is the way that we control our enzymes so that we don't waste energy making things we don't need. Finally, we went over a bunch of conditions about what will affect our rate of reaction. So we have all these graphs about reaction rate. And the idea is that as we change the conditions, we can make the reactions go faster or slower. Enzyme concentration and substrate concentration look very similar because in general, if you add more enzymes, you should get more activity. The more workers you have, the more you can get done. However, it does start to level off, and this is the part that's important because at some point you run out of stuff to work on. This is the whole like too many cooks in the kitchen idea, where if I have, say, 5 ovens for some reason, and I guess I would have a restaurant probably, if I have one cook that's going to get me some food. If I have 2, that's more. 3 more, 4 more, 5, yeah, more. But then I get to 6, and that 6 isn't going to help me that much, and 7, and if I keep adding on top of that, it's not going to help that much, right? And so adding more and more enzymes at first will help, but then it will level off. Same with the substrates, right? At first, if I add more and more stuff to work on, let's say in this case I have one chef, and the chef is trying to cut like carrots or something, and if I give him or her I don't know, like 1 carrot every 10 seconds, like the chef can get a certain amount of work done. But maybe the chef is sitting there waiting for me to give the carrot, and so then I can give 2 at a time, and the chef can go faster, and then I'll give 3 at a time, and the chef can go faster and faster. But at some point, the chef's going to reach its maximum carrot cutting speed. And if I start dumping 10 and 20 on the chef, then this is not going to add any speed, right? And so at first, adding substrates is going to make it go faster, but then after that, it's not going to help that much. Temperature was an interesting one. Temperature there's 2 things going on during temperature. The first is the idea that raising the temperature makes reactions go faster, right? This is kinetic energy. This means that, to think of it from the opposite perspective, if I make the temperature lower, the reaction is going to get slower and slower as the molecules move slower and slower. But once you reach a certain point, the energy becomes so great that it starts to break the protein. It denatures the tertiary structure, and the enzyme is going to unfold. And that's going to make the active site not work. And then it doesn't do any reactions anymore, right? And so there's an optimal temperature for enzymes. The optimal temperature is dependent on the species. Most of the time, you're going to get something that's right around normal room temperatures, like humans and things like that. But you should probably pay attention. That might be a way that they might try to throw a little curveball in there by changing the species or changing the temperature. pH is like temperature in that there is a ideal value for it. It does depend on the situation. Evolution has made enzymes evolve to the environment that they're most found in, like acidic situations in your stomach, or basic situations like in your intestines, whereas most of our situations, of course, are around 7 and neutral. But of course, this is all about adding or removing hydrogen ions. If you put it if the enzyme goes in something acidic, there's tons of hydrogen ions floating around. Those are going to disrupt the hydrogen bonding. Of course, if you put it in the base, it's going to start stealing hydrogen ions from the enzyme, and this is going to disrupt the hydrogen bonds and stuff like that. So we're going to get a denatured, no matter what happens, going high or low, the enzyme will denature. Finally, salinity is a lot like pH because salinity has to do with ionic concentrations. At some point, if you just have so much sodium hanging around, the sodiums are going to start attacking things and stealing parts of your molecules. But at some point, we do need a certain amount of salinity, which happens, again, interestingly, to be right around the salinity of the ocean. Because of course that's where most things evolved way back then, right? But if you add ions or remove ions, then this is going to affect your enzyme, right? So there you have a very quick review of enzymes and energy. Let me know if you have any questions. Go ahead and try the practice quiz, and then if you feel comfortable to do the progress track, go ahead and do that. But again, like I said, hit me up on Google Hangouts or whatnot if you have any questions, comments, or concerns.